to basically introduce this concept of recombinant inbred lines, which has been talked a bit by the lecturers in this school so far. But how do we construct it? I'll briefly go over it. Then what I want to present is basically the derivation of a result for two loci by Haldane and Waddington in 1931. And because I'm going to present the derivation because that is the derivation I'm going to extend for any loci. And I'll try to explain what was the long-standing problem which stood there for about 80 years, which people were unable to solve, that we have solved. So I think my main goal is to basically convey the problem rather than spend a lot of time telling you the solution, because the solution is all there in my paper and the supplementary material. But I think it's very important to understand the problem. So I have made a conscious choice basically to convey the problem rather than giving you the solution in detail. So because that's very simple. And furthermore, the math involved is very simple. There are a lot of assumptions, but I think on the board I have written it. I have it on the slides as well, but I thought it is much more clear if you go through the math on the board actually. So with that, some of this will be repetitive, but it is good because you'll know what I'm trying to do. So if you look at sexual reproduction and meiosis, basically if you start with two homologous chromosomes, there is an exchange of genetic material through crossover. And this crossover can lead to formation of non-parental or recombinant gametes. Okay? One should remember that the probability of recombination usually increases with the genetic distance between any two loci. And furthermore, the work that I'm going to present, I will be always displaying it with just one chromosome because I want to put two linked loci rather than start with two different chromosomes where things are independent. Okay, so in all my slides, I will be basically showing one chromosome, one pair of chromosomes, but it all applies to other few things are on different chromosomes as well. But then they are independent of each other. Loci are independent of each other. So just to make it clear, if you have a heterozygous state here, after meiosis, there are basically four possible gametes that can be formed. Two which are of the parental type, and two which are of the recombined or non-parental gametes. Okay, if you say that the probability to have an odd number of crossovers between this interval between the two loci is r, small r, then the probabilities of these gametes are given as follows. Okay? Here I'm assuming that one, I'm taking the Haldane model where crossovers arise independently. Two, which is true, is that you need odd number of crossovers to basically get a recombination event between the two loci. If there are even number of crossovers here, then you won't get a recombined gamete. Okay, these are two simple things. So these are the frequencies of these gametes that you expect, and the way to estimate is basically you can compute the or you can determine the number of recombined gametes and the non-recombined gametes, and use this formula is the estimation of the small r, which is basically the probability that odd number of crossovers occur between two loci. Then I would like to describe basically what are recombinant inbred lines, the RIL for simplicity. And there are two ways to produce it. One in the case of plants where you can do self-pollination, which is the selfing. And today's talk is basically all the results are about the selfing case. But I think what we have been able to do is that we have been able to also extend it for the SIB case also, which I will explain in the next slide. So what are recombinant inbred lines? They are basically strains that are produced through repeated inbreeding, and they constitute a stable and permanently shareable genetic resource. But how do you achieve that? At the F0, like if you remember the Mendel's case, you start with two homozygous parents, which are shown here, and you cross them. In the next generation, recombination or no recombination, all the individuals that you expect are of the heterozygous state. That's what you'll get. Even if recombination occurs here, you will get the same gamete. Okay, is that clear to you? From F2, what you do is you pick a single individual and do self-pollination for a number of generations. And the idea is to go till infinite generation or to fixation. Okay, and if you do that, you basically eventually will get a fixed state, a homozygous state. But the final strain that you will get will be a combination of the alleles that you started from the two parents. Okay, So basically, you are doing it for one individual at a time. Similarly, you can start with another individual and you will get a different strain at the end of the day, which is a fixed state as well. And essentially what you will get in this process is a mosaic of the parental strains at the end of the day. These are the recombinant inbred lines. I'm not an expert on that, but I think what I've been told is that it's useful both for genetic mapping and to identify genes contributing to both traits of interest in agriculture and disease as well. So this is how it occurs in plants where you can do self-pollination. So you can do a selfing here. In the case of animals, what they do to construct the recombinant inbred lines is basically they start with again two homozygous parents, they cross them, but in the subsequent generation they basically cross brothers and sisters. And this is perhaps the process which has been used a lot to construct the mouse recombinant inbred lines. So that's about recombinant inbred lines. And the work of Haldane, which we want to extend, which basically appeared in 1931. Actually, the formula appeared even in a previous work of Robbins in 1918, which we discovered actually while we were writing the paper. But their derivation was slightly more difficult, so they did not attract that much attention over time. But the formula is basically, it asks the following questions. So the point is that at the end of these steps, what you have are the fixed genotypes if you go to infinite generations. So if you start with two loci, and the example is shown here, you start with two loci, you start with two homozygous parents. In this F1, if you cross them in the F1, you get the heterozygous state. And from there, you can follow different individuals. And finally, what you expect are a combination are the four genotypes. These are the parental genotypes that you started with and the recombined genotypes. There are four possible genotypes at the end of the inbreeding process at infinite generations, if I say so. So what people are interested in is to compute the frequency of these recombined genotypes. What do you expect at the end? So the way to compute it is that you get the number of recombined genotypes and you get the number of parental genotypes, or the basically the number of recombined genotypes by the total genotypes gives you the probability of getting a recombined genotype. No mutation, no selection. I always forget, so I had put it here: neglect mutations and no mutation, no selection. That's very important in this picture. So the work of Haldane, in summary, what he did was that he related this R, which is the big R, which is basically the probability of getting these two recombined genotypes, to the small r, which is the probability of getting an odd number of crossovers between any two loci. Okay, so and the result is this: capital R is equal to 2 into small r by 1 plus 2 into small r. Okay, and basically I want to spend about 5 minutes going through that derivation, which is not actually the original Haldane derivation, but a simpler derivation of that. So please remember that what you expected at F2 are the number of genotypes that you expect at F2 are actually 16. Okay, and to be very, very precise, if you have L loci, the number of genotypes that are possible is 4 to the power L, and the number of recombinant genotypes, the number of fixed genotypes are 2 to the power L. Okay, I'll make it more precise later on. But let's go through the derivation of this formula. Capital R equal to 2 into small r by 1 plus 2r. Again, I will like to specify that this is not the derivation which was in the 1931 paper, but is a much simpler derivation which has been figured out recently. So what you do basically in the generation of recombinant inbred lines, to recap, we cross the homozygous parents, we get a heterozygous at F1, and then we start with one individual and continue with it with self-pollination. At the end, you will expect to get one of these four. Okay, those are the surviving genotypes at the level after a lot of inbreeding. So basically, with two loci, there are, as I said, 4 possible states at each loci, and there are 4 square equal to 16 F2 genotypes possible. One can easily compute the probability of getting each of the F2 genotype. For example, this is an F2 genotype whose probability is given by the product of getting each of these two gametes, and it's equal to 1 by 4 into 1 minus r by r, okay, into r. So let's go through the derivation, which is better on this board. I'll try to simplify it. So you start with, to recap, you start with the two homozygous parents, you cross it, you get the heterozygous. Here the probability, you have only one genotype, no matter what you do. So at F1, you have only probability of 1 for this genotype. At F2, you will get 16 possible genotypes. And the 16 possible genotypes are written in the form of the Punnett square here, and the probability are written here. What you can do is, to simplify this problem, is to classify the genotypes into 3 classes. So the first class is that where both loci are fixed. Okay, those are the final genotypes that you expect. So by symmetry, so these are the 2 parental types. So their probability is same, and you can say that if the final probability of the recombined genotypes is capital R, the probability of getting any of these 2 genotypes at the infinite generation is 1 minus capital R by 2. Okay, and any of the recombined genotypes is capital R by 2. Okay, so basically, if you look at the diagonal elements, the 4 genotypes, these are already fixed at the F2 generation. Subsequent inbreeding will not change anything because we have neglected mutations. Those have already gone to fixation. Okay? The second class of genotypes are those, for simplicity, which have no fixed loci. Okay? So they are the ones which I have written in blue. Okay? So there are 4 of those, and actually you can convince yourself that both these genotypes are equal to the one you started with F1. Okay? This genotype and this genotype is the same as F1. So the result finally, if you start with that genotype with at F2, will be similar to the one what you would have got if you had started with F1. Okay? So I'll try to make that formula clearer now. And the third class of so there are 4 such genotypes which are fixed at no loci. The third class is those the remaining 8 genotypes which are fixed at just 1 loci. Okay, I will leave you as an exercise, but tell you that if you start with any of these 8 genotypes, in this finally what you will get is that 50% will go to the parental type, 50% will go to the recombined type. Okay, you believe me on that one. If it's not clear to you, I'll be happy to show you my notes and make it clear after the talk. So there are these 3 classes of genotypes, and one can easily specify them with this M, which denotes how many loci are fixed out of the 2. So now what we want to do is basically express the capital R, which is the fraction of recombined genotypes, in the form of the small r. So what we do is that we try to see if you start with these 16 F2 genotypes, what will be the eventual destination, or what will you eventually get outcome of this process of inbreeding. Okay, I hope I've convinced you that if you start with these recombined genotypes already, which is at fixation, okay, with both fixed loci, you will get only subsequent inbreeding will only give that only. So you sum r square by 4 and r square by 4, these 2 genotypes, r square by 2. So m equal to 2, these 2 genotypes will contribute directly to this r. Okay, these all go to r in subsequent generations. So there are 8 genotypes here, each with probability r into 1 minus r by 4 with 1 fixed loci. So if you multiply by 8, it becomes 2r into 1 minus r. As I said, 50% of that will go into recombined types, 50% into non-recombined types. So I take that quantity and multiply by half. That's the second contribution to the recombined generations at the infinite generation limit. And the third is basically the class of genotypes which are fixed at no loci. Okay, so that for that you need a symmetry argument. I hope I have convinced you that these 2 genotypes which are fixed at no loci are the same as the F1. Okay, sorry, same as the F1. So the outcome will be the same. Okay, so we said that if you start from F1, the fraction of recombined genotypes is capital R. Okay, so if you started with 1 minus r square by 4, which is into 2, 1 minus r square by 2 of these 2 genotypes at F2, the fraction of recombined genotypes at the end would be 1 minus r square by 2 into r. Okay, right? Is that clear? And the third class, which is slightly difficult to understand, is basically if you start with these 2 genotypes, which are fixed at no loci, but the gametes are the non-parental types, the recombinant types. If you think in a symmetric argument, if you start from here, the parental types are the ones which are the recombined types. Okay, so it will be 1 minus capital R times the probability here. So that's the last term. Okay, So you have capital R on both sides, you can solve and you'll get R equal to 2R by 1 plus 2R. That's a simple derivation of this Haldane-Waddington result. So it was easier to do for me on the board actually rather than the slides, I must say that. So the main point is that uh, you can relate the capital R with small r using this Haldane-Waddington result, and this result has been used for genetic mapping in GeneMaker and many other softwares. Okay, that's what is used. What you can do is that you can then ask, can we do this for 3 linked loci? Can we compute the probabilities of the final RIL genotypes if you are following 3 loci? Okay, and people have done that. So in the 3 loci case, what will happen is basically you will start with 2 homozygous parents. You have 3 loci here. Final outcome will be basically 2 to the power 3 equal to 8 possible RIL genotypes. To be very specific, what we want to do is we want to compute the probabilities of these 8 genotypes at infinite generation limit. Okay. So, but again, one should realize is that there is this symmetric property where the probability of this genotype is same as this genotype. So actually, rather than computing for 8 unknown probabilities, you actually can compute for 4 unknown probabilities because of this symmetry. Okay. So it is like this. So if you see, the probability of this genotype was equal to this genotype, the parental types. Okay. So similarly, there are 8 possible RIL genotypes. You can condense them into 4 unknown quantities. Yeah. So that's the first thing. So to solve for 4 unknowns, you need 4 equations. And one equation clearly comes from that the sum of the 8 probabilities of these 4 unknowns is equal to 1. That gives you one equation. Okay. We need 3 more equations to solve this system. And those 3 actually equations come from actually if you ignore one of these loci at a time and compute the small r for those 2 loci. So there are 6 such possible small r's, which basically tell you what is the probability of getting an odd number of crossovers between any 2 loci. Okay, so you have to look at loci only at pairwise. Okay, so there are 6 such possibilities. Sorry, there are 3 such possibilities. I'm sorry. So these will give you 3 more equations. Okay, so you have the equation, one equation for the sum of the 4 probabilities giving 1, and there are 3 equations coming from these Rij's. Okay, and that gives you 4 equations, and you can solve this system. I'll try to make it much more clear in the next slide how you do it. So you had these symmetries giving you basically the 4 unknowns. These the sum of these is equal to the capital R12. You can convince yourself, and then you can write 3 equations based on that, and you have this sum of the probability is equal to 1, and that gives you very simply the solution for each of these 4 unknowns. Okay, so you can solve it. So basically the main point is that if you know the 2 locus probabilities, that is R12, R23, R13 from the Haldane formula, you can actually directly use a simple extension and compute the 3 locus RIL probabilities. Okay, that was also shown. The point is that can we use this trick to go to a next level and compute the probabilities of RILs with 4 locus? That's what we wanted to do. And actually, I'll tell you what, when we did it and sent it to most journals, the referee said that that trick works without even computing, and you have done a trivial job. So actually, no, you use this formula to get the probabilities each of the 8 genotypes. Sorry. So there are these 8 genotypes probabilities, and you use the 2 locus results to get that probability. Okay, is that clear? Or I should expand. Okay, so the point is that if we go to 4 loci, can we use this trick? That is, what we have is enough to solve that system or not? That's the main question. Okay. Yes, if you don't ignore one of the loci and we're just computing the recombination between 2 loci. They are. But it's easier to find those numbers than maybe the 3 loci numbers. So the point is that can we use this R12s and the 3 locus result and the 2 locus results to get the 4 locus result? Okay, it's an analytical solution. Do we get an analytical solution or not? So again, for 4 loci, let me convince you that there are 16 fixed genotypes. Okay, and again due to the symmetry, there are they come in pairs, so you have 8 unknowns. Okay, To solve this system, you need 8 equations. One equation clearly comes from the sum of probabilities being equal to 1. So you need 7 quantities, okay? 7 more unknown equations, 7 independent equations to solve the system. 6 of them can come from these, again, these pairwise recombination rates, okay? The RIJs. So it gives you 6 equations like the previous case of 4 loci where we got 3 equations because of that. So you want one additional equation. So there are 6 from the RIJs. There is one additional equation coming from this probabilities being sum of probabilities being equal to 1. So we need one more equation. So it's tempting to use one of the 3 locus uh, you know, uh, relations to solve this system. But actually, if you think about it, the 3 locus result was obtained as a result of 2 locus result. So though that equation, such an equation will not be independent. And actually, if you solve this system with a 3 locus result, you get the rank of the matrix to be equal to 7. So this is not possible with that. So there is one missing quantity for the 4 loci, which is not there in the not available from the 2 locus and the 3 locus results. So how do we solve this system? So that is the problem which was standing, long standing, if I say so, for 80 years. So because you could not get a exact analytical solution for the 4 loci case with all the assumptions in this model. Okay? So that's how, and basically the work is about how do we solve it. But let me, before going that, try to generalize this problem a bit more. So in general, one can ask, what is the probability of RILs with L different loci? L could be 2, 3, 4 and higher. So with L loci, you have basically 4 to the power L possible genotypes. The number of RIL genotypes are 2 to the power L, which are fixed at each loci. Basically, we want to compute the probabilities of these 2 to the power L genotypes. But as I mentioned, due to symmetry, these genotype probabilities come in pairs. So actually, the unknowns become 2 to the power L minus 1. And however, the set of RIJs, the probability of getting crossovers between pairs of loci, the number of them are L into L minus 1 by 2. As soon as L becomes greater than 3, 2 to the power L minus 1 is greater than L into L minus 1 by 2 plus 1. Okay? The 1 being for the plus 1 being for the sum of the probabilities of all genotypes being equal to 1. Okay? So as soon as you go beyond 3, there's a problem. You need an additional equation. I would like to give you the result now because it will get slightly more technical after this. Is that from 3 to 4, you need one additional quantity, which our framework will give you. From going to 4 to 5, you don't need additional quantity if you know the 4 locus result. But from going to 5 to 6, you need one additional quantity, which again our framework will give you. From 6 to 7, you don't need additional quantity. Whatever you have till 6 is enough. From 7 to 8, you need additional quantity. In general, our framework is able to give a solution for any number of loci. Actually, we can write down on paper and pen results up to 10 loci, but we have a computer program which basically does it for about 20 loci very easily. One important point, you may ask, if we start from F2, we know what is the probability at F3. We can just do this with a computer program and try to solve it. But remember, we want the probability at infinite generations. As L is becoming larger, this is exploding. So such a brute force approach, if you write a program, basically you can do up to maybe 6 or 8 loci at best. But beyond that, it becomes infeasible. So you need an exact analytical result or you need to approximate. So that's the difficulty. That's why we are looking for an exact result here. Yes, so in principle, if you go to 12 or 13 generations in a computer program, you get it. But if you are willing to do that with a reasonably, let's say, a i7 computer with a brute force, we are able to go to 8 loci. So we don't go to infinite loci to measure that, but let's say you go to 12 generations to approximate that. I'm saying that that is doable in, let's say, 1 day for 8 loci. Okay, so and in I would say 2 hours, our framework, our computer program will give you for 22 loci the result. So with this framework, there are various simplifications we have. One finer point, because you may not follow beyond this because it will become technical, our method is that this method, until now what I have shown, it really is computing these probabilities using the Haldane model of crossovers, which assumes that the crossovers fall in an interval independent of each other. But in reality, there's interference where there is a correlation between the first crossover and the next crossover. Okay? And there are detailed models to model this crossover interference, if I say so. The framework that we have actually allows us to also compute in the presence of interference. Although it's a computer program, but this allows it pretty fast to do that as well. This is not shown in the paper, but I can tell you that this also allows that. So with that, basically, let me try to first give you a recipe of our method, and then I will try to illustrate the method using the 4 loci case. So what we do is basically we use some notation motivated by physics to denote these, RILs. I'm sorry, I'm not even competent to speak about spin glasses. Kavita is better. But huh? Yeah, so I will be honest. I was telling Rahul that the idea that it could be done using these 2 simple theoretical physics equations was my advisor's. My contribution really is basically that I got a lot of simplifications, which throws out a lot of computations which I ideally would do. So, and there's a very elegant approach of the trees which I came up with, which really allows it to solve very nicely this problem. So, of course, again, this idea that you can do it this way is certainly not mine. I'll be very honest about it. So, what we do is that you can try denoting the homozygous state small a by small a at a given locus by the spin variable +1, and the capital A by capital A by spin variable -1. So if you convince yourself, if there are L loci, there are 2 to the power L RIL genotypes, and in the spin notation there are 2 to the power L states. Okay, and basically what we are interested in is to compute the probability of each of the 2 to the power L states. Okay, but we should remember that because of the symmetry, it's actually two genotypes always have the same probability. So basically the unknowns are 2 to the power L minus 1. But I won't bring that complication in for now. So what we do is that we basically ask that what is the expectation of these different k-spin products, and if we are able to compute these expectations by some chance, some way, then they will give you the probability of the states. Okay, and for the record, the number of k-spin products starting from 0 to the number of L loci is equal to the number of RIL genotypes, 2 to the power L, if you sum them up. So again, each step is done in very detail without missing anything in the supplement of this paper. So you are welcome to look at it if you are at all interested. So let's say we are able to compute these expectations. How do we go from these expectations to the probabilities of each of the RIL genotypes? And that's given by a formula by Roy Glauber, which we exploit, which is the following. Okay, so if you ask the probability of this RIL genotype, so let me try to make it slightly more specific. So which one? It's a spin flip. I'll show you. So I actually have done the 2 locus result there with the spin notation, and to get the expectation here, which I will come to. So I hope I have convinced you that so let's say this was the RIL genotype. We just considered 2 loci. What this says is that if you were to look at this probability, this is actually the first in the spin notation. It is +1, -1. Okay, fine. And the way Glauber's formula will give you is basically you put E 1 plus. So this is +1. So S1 is +1 into S1 by 2. Yeah. And 1 plus minus S2 by 2. Okay, is that clear? So if you expand this, you will basically get the probability of each RIL genotype in terms of these expectations. Okay, if you have all the expectations till L loci, all the spin products, then you can basically compute very easily using this formula back the probability of each of the RIL genotypes. So let me tell you here, if you don't understand beyond this, is that that missing quantity for the 4 locus case is basically the expectation of the 4 spins, basically. If we can compute that, we can solve that problem. Okay. For the 6 locus case, again, if you have all the 4 loci results, then you can just compute the expectation of the 6 spin products, and that will give you the answer. Yes.
But the expectation of that precedes the correlation.
Yes. So again, I was telling Rahul, my colleague here, that I don't completely understand that, but I will try to explain you how that. So maybe I'll try to concretize the question, which might interest you. That this is very critical, because if this was not the case, we won't be able to solve using this framework the probability of RIL genotypes under the crossover interference.
Because the uncorrelated, you have a Haldane model.
Yes. Yes. Yes, but I think this expectation holds regardless of these spins being correlated. That's why it works. But actually, the probability of the gametes, though, which will come in the next formula, they don't hold. So that needs to be computed with the correlation. And that for that, we need to run a specific model for crossover interference. Okay. But that makes really life easy. That's why the framework works even with crossover interference. So, so that's the first thing, that we exploit the Glauber's formula to go from basically expectations to the probability of the RIL genotypes. And the second part, which again I'm not competent enough to speak about really, there are many people in ICTS who would know about this much better, is basically we use the Swinger-Dyson approach to basically relate a higher order spin product in terms of itself and the lower order spin products. I will basically illustrate this in detail for the 4 locus case. Okay. And the step 5 is that one of the simplifications you should note is that so you have all the spin products up to loci L, but you should note that all the odd spin products vanish. Okay, only the even spin products remain. Okay, and for the 4 locus case, which I am interested in, we already know the spin product for the 2 loci. Okay, 2 order spin, 2 spin product, and it's given by this formula. But let me just go through that, because that will be very important for the deriving the 4 locus case result. So basically, if you had these 4 genotypes, which are RIL genotypes, which are fixed at infinite generations, you can write them in the form of the spin notation here. The sum of the probabilities of the 4 genotypes will add up to 1, we all know that. But the expectation of the 2 spin product is written as follows. Okay, so and these are the parental types, and these are the recombinant types. So these you expect at probability 1 minus r, this as probability capital R. If you add these 2 equations, you get the expectation of 2 spin product as 1 minus 2r. Okay, so that's the result we'll also exploit for the 4 spin product case. Good. And finally, after you get these expectations, you have computed these expectations, you will use the Glauber's formula to basically get the probability of each of the RIL genotypes. That's broadly the framework. And the reason we present it in this framework rather than showing the analytical part is because this recipe works both for the no interference case as well as the interference case. Okay, so what I want to do is basically, I want to illustrate briefly the method which we use to solve the 4 loci case, and this can be extended to 6 loci case, which has been done as well. Yeah, so okay, let me just draw something, and that will maybe clarify. Okay, so is that part visible? Okay, so okay, so I think it will get clarified in 2 slides. I think allow me that. So I think it's better to do that. So basically, the point is that in what Haldane assumes to compute this quantity small r, which is basically the probability to get odd number of crossovers between any interval or any two loci. He assumes that the crossovers fall at random. But in reality, what is seen is that if there is one crossover, then the next crossover has some correlation in the genome, and there's some interference. Okay, and this people have understood, and there are detailed models to exploit this. So what I want to say is that this framework, although I'm showing it for the very simple case, it actually works in that detailed case as well. Which one? They're not correct. I think I wanted to make simplified. So you can imagine all the spin products. Uh, Actually, I can tell you that, allow me till end, I will tell you that even all the two spin products don't survive. There are only very specific ones which survive, which have a certain property. That also helps us speed up our program. Okay, so basically what we want is this 4-spin product, and actually the Swinger-Davidson approach allows us to write it in terms of itself, the 4-spin product, and the lower order spin products. And actually there are 6 spin products, if you can imagine, between, if you have 4 loci, yeah, 4 into 3 by 2, so, and only 2 of them contribute. I won't have time here to explain why only 2 of them contribute, but what I want to stress is that I showed you the derivation there. These 2 spin products, we know the formula. So if you can directly plug it in there and solve this, and you'll get the 4-spin product. But I've not yet told you how you get this 4-spin product, right? But again, the Glauber's formula which I showed you, the simplification for the 4 loci case is this one. This will, again, if you know these spin products, the 2 spin product and the 4-spin product, you will get back the probabilities of the RIL genotypes as well using this equation. Okay, so I think the key to this method is as follows. So it's actually illustrated here itself. What I did to solve this case in some sense is that what we do is that we ask the following. Let us classify, starting from the F2 genotypes, which are the real, what are the trajectories that will lead to the different real genotypes. Okay, and one can then write the expectation in terms of the expectations at the F2 state, which is this is the expectation at the F2 state. I know this is very difficult to grasp maybe the first time. And here is the formula, which is the expectation of the spin product, and these are the probabilities of getting the 2 gametes that form the RIL. Okay, and to make it very specific, here, as I shared in the Glauber's formula, their spins are independent, but here, you know, the interference will play a role when you compute these probabilities. And actually, what we know is that there is a simple trick to go for from the expectations at the of the F2 genotype to the expectation that you would have if you started with the same genotype from F1. And I will just go over the simplifications, which is the following. If the F2 genotype, if the locus K is of this form or this form, then you replace the spin by 1 and -1 in this case. That's what you can do to relate the expectation at F2 state to the expectation at the F1 state. Similarly, if it's of the heterozygous type, you can just replace the spin by the same spin. But however, if you have a locus in the F2 genotype of this form, then actually the spin is replaced with a negative sign. I won't have time to go into the details of motivation for this, but you're welcome to read it if you want to do that. But if you apply these substitutions on the this part, the right-hand side, then this expectation can be written in terms of the expectation at the F1 state, and that allows a self-consistent equation which allows you to solve the problem. A trick that we use to compute this quantity, which is very important, which multiplies each of these expectations, is mapping these different genotypes that contribute to a tree-like approach. And actually, what we find figure out later is that there are several trees we know through symmetries which will not which will not contribute. So we are able to exclude them and only compute for the trees that contribute to the equation. So let me try to illustrate you the quantity that will multiply this 4-spin product in this equation, this quantity. Okay. How do we get this quantity? This quantity is given by all the genotypes that are heterozygous at each of the 4 loci, and these so there are 16 such genotypes, and due to symmetry, again, you can just consider 8 of those and multiply it by 2, and the tree is shown here. Okay? And it comes with a sign. And finally, over if you sum over the 8 genotypes, you get this quantity. Back to the question of interference, right? If there was interference, we won't be able to multiply this. That is, the probability of this won't, you cannot, these are not independent, okay? So there, what we do for the crossover interference thing, we have a model run over it and just compute that one. This is very elegant to give an analytical solution when you have no interference, because you can just, these are independent of each other, you can multiply them in this equation. Okay? So that's where it is. So, and so this basically is multiplied by 2 and divided by 4, because there is a probability of 1 by 4 of having this, this allele state at the first locus. So that's what we do. You're welcome to read in detail in the paper. For the 2 spin products, if you remember correctly, so for the 4 loci case, the possible spin products are 0 spin products, 1 spin products, which will contribute 0, as I said, odd spin products, 2 spin products, and 3 spin products, and 4 spin products. The 3 spin product again is of odd number, so it will contribute 0. There are a number of 2 spin products, but only 2 of them contribute. You are welcome to read again the paper to figure out why they don't contribute. But the 2 which contribute, again we can use this tree approach to sum over them and get the quantity that multiplies the 2 spin products. Okay, this is the quantity. And finally we basically combine all this to get this equation, and this you can solve to get the unknown quantity, the missing one equation which is required to solve the four loci system. Let me tell you a bit about the simplification and I can conclude. So which are the states which will not contribute? So in the no interference case, some of the nice things which we found is that if genotypes have this structure, so let me specify. So for the 2 spin products, the genotypes that will contribute are the ones which are heterozygous at 2 loci. But if the genotypes are such that they come in this kind of structure, they're heterozygous in the first loci, homozygous in the second, heterozygous in the third, homozygous in the fourth. So they come alternate. Such spin products don't contribute. They basically sum up to 0. The only ones which contribute are the ones which have these come in pairs actually. So there's a detailed explanation on that. And this is again very important because realizing these symmetries, and that this argument actually holds for even higher loci. This actually allows us to just discard a lot of genotypes, computation over a lot of genotypes. We are only able to compute only very small fraction of it, so that's why we are able to go to about 22 loci. So to sum up, I'm no expert in RILs, but I understand that they are important to map both molecular markers and associate traits of both agricultural importance and diseases with 2 chromosomes. This problem of Haldane has resisted extension to 4 loci for a long time, but it seems that nobody had tried these very simple physics approaches to solve it, and we believe that these approaches can really help other population genetics problems. I'm not aware, I do not work there, but I think this approach can actually have impact in other population genetics problems. Furthermore, we basically have shown that this simple tree structure that we have to compute these probabilities allows us to compute factors for any number of loci and also allows a lot of simplifications. And this basically we have incorporated in the computer program to basically compute the probabilities of RIL genotypes for any number of loci in the no interference model. And also we have a program which we are going to publish soon, which will do it for the interference cases. With that, I would like to thank again my postdoc advisor, former Olivier Martin, my close collaborator for this work, funding which basically allowed me to do this work, and present funding which allows me to do research today in India. Thanks a lot for your attention. Happy to take any questions.
Can you get a closed form solution to multiply with interference?
Yes.
Write down an analytic solution.
I
That lets you help you fix the problem. You handle a loci which are all are no interference.
Means you have to specify an interference model first.
Sure, but you write down an interference model between all the triplets that you reach along, write a probability model that can you use method based those formulations. That would be a big advance. That's I think.
No, I think three loci case interference. Oh, okay. So you're saying that.
Something to think about. I mean,
No, no. I have a feeling that I don't work on this, and my former advisor works a lot on maze meiosis. I think he has worked on this. I'm sure he has computer programs to do it. But do you think the analytical solution would be a big advance?
oh, it's all about how it works. I would.
None of the referees said that. So, to be very honest,
Genetics, not physical literacy.
no, no, we went to all the biojournals first, and so it was the main problem we had, and we basically got in, and one of the big places, I don't want to name it, was that I would say there are only 3 or 4 experts on this, and one of the experts said that this can directly, the 2 locus result gives you the 4 locus results. We went back with the math, and the editor said, I can't understand. So I believe you, I believe also the referee. So yeah, so that's a good point, but I think there are some results, but I think I can get you that if you want. I think I've gone over time if you. Physical review letters. Yes. You mean that's where all this is published? Okay. Yes. Yeah. Thank you very much.